We're at the 25th CROI. We're here with Jonah Sacha, and he is a researcher at OHSU, Oregon Health and Sciences University in Portland, Oregon, where we're from as well. Yeah. We don't see each other there, but we see each other at the conference, wherever, Kuala Lumpur, wherever it is. But you've been doing a lot of science at OHSU, and we thought it might be good just to talk a little bit about the work that you're doing.
Sure. So would you like me to discuss the CMV vaccine vector that I presented here?
Yeah, I think that'd be great to start out with. Yeah.
Okay.
Yeah.
The CMV vaccine vector, which has been used exclusively to date in rhesus macaques, is the reason I moved to OHSU, because it remains the only vaccine that we have for HIV to show really stringent protection. And the backstory of this is that when you vaccinate animals with this vaccine, about 50% of them will spontaneously control viral replication, and if you follow them over time, they will actually clear the viral reservoir, and they're indistinguishable from animals that have never seen the virus. So that's just stunningly amazing.
Right.
And it's all predicated on the use of the cytomegalovirus, or CMV, as a vaccine vector, and it has a very unique characteristic, which is once you're infected with this virus, it's a subclinical simmering infection that's persistent for your entire life. And we think that's the key to what makes it work so well.
Is there any, why would it contain so well in animal models and not in humans? Is it there's more reservoirs, or what would you think would be the challenge?
So it's the CMV vaccine vector has been all preclinical to this point. Bill and Melinda Gates Foundation has given a large amount of money to move it into clinical trials, and those are actually going into clinical trials in Portland at OHSU, and it's planned to happen at the end of next year, so in 2019. This is a phase 1 trial, so it's predominantly safety focused, and so there won't be any, this is in people who are low risk for HIV acquisition, but I think that when it proceeds to an efficacy trial, I think it will show similar efficacy.
Oh, really?
I do.
Yeah.
And the reason is that we've now been able to recapitulate the unique features of this vaccine in a second non-human primate species. And so the vaccine vector, which is human CMV, has been genetically modified to resemble those viruses. And so I think it actually will be efficacious in humans.
I'm trying to think where the last presentation was given on it. Was it Lewis Picker did it in Vancouver, or wherever it was? He gave the last presentation of the conference. Everybody was still there. You know how that is.
Yeah,
where it is. Okay. So that tells the story right there.
Yeah.
But it's a great opportunity. So what else is it that you're working on? Is there other projects?
It's quite a few things. So we have quite a wide breadth of stuff going on now. The thing that I'm most excited about besides CMV is our work with allogeneic stem cell transplantation. And this is all work based on trying to determine what functionally cured Timothy Brown, aka the Berlin patient. So as you know, he was the only person to date who's been functionally cured of HIV, and it was because he received an allogeneic, so from a stranger, a bone marrow transplantation.
So he's now a different person, essentially,
right.
Genetically.
He has someone else's immune system grafted into him. And he hit a remarkable mark last year. He hit 10 years of drug-free remission with no signs of HIV rebound. It's very difficult to prove it negative. The old axiom that absence of proof is not proof of absence, but this gets about as close as you can.
But 10 years is, I think the others, the Boston patients were like,
they relapsed in a year. So, the question remains: we still don't understand the mechanism of how he was functionally cured, and it's difficult to do that in the clinic. You mentioned the Boston patients; they rebounded, so we still don't understand this, and so we actually decided to build a preclinical non-human primate model of fully MHC-matched allogeneic stem cell transplantation. So, in order to do this, I went to the bone marrow transplant department at OHSU and spent a lot of time with the physicians there. And learned about their excellent care and how they do this in humans, and we mirrored exactly what they do in humans with non-human primates. And to date, we just published a manuscript in Nature Communications where we showed that we can get full engraftment, so we can replace an animal's immune system with that of another animal. Wow. And we're doing studies right now in animals that are SIV infected, ART suppressed, and we have a current animal named Beck. She is 36 days post-transplantation, and she has 96% T-cell engraftment. So when that gets to 100%, what's going to happen with the reservoir?
Yeah, right. And the reservoirs are always, and then maybe it'd be a good time to really explain a little bit about the reservoirs, because they're not really it could be the skin, it could be an organ, it could be...
Right, people have found latently in HIV-infected cells everywhere.
Everywhere, every cell in the body.
Well, not in every cell in the body, but we found infected CD4 T cells in adipose tissue. We typically think about it being concentrated in the lymph nodes and the gut. But you can find it in the kidneys, you can find it anywhere the CD4 T cells are. We find
and those areas are quiescent, they don't seem to do much, or some of them.
We don't know. Yeah. All it takes is one cell to reactivate in the absence of drug, and you can spark the infection all over again.
So it's an amazing thing. I think the more I hear from you guys at OHSU right here in my backyard, we ought to talk more, but I would really like to do, if you do any talks up there or other, because we do video all over the country.
I'd love to. We do a lot of outreach too. That's the thing that I'm most proud of, is that we bring the public, and we also bring a lot of middle schoolers and high schoolers and students into campus to show them the research that we do, and show them we work with animals, this is the care we give, and this is why we do what we do.
Right. It's critically important.
It is.
For mankind to have this opportunity to fully research what we need to do, and easy enough to explain why.
Yes.
Because if we get an answer, every answer is good, because you know what you either know or what you now know that you are not going to pursue.
Yes.
And what's the other axiom is that one positive answer creates 20 more questions, and
that's exactly right.
It's crazy. And now you have to decide what are those 20 questions we can actually afford to answer or fund to answer. So it's a challenge all the way.
Yeah. One thing that I do want to tell you about this, it's the final piece of the stem cell transplant project, is Timothy Brown's transplant came from someone who was CCR5 deficient.
Right.
And CCR5 is the major co-receptor for HIV. And we think that played a role. We're not entirely sure how significant it was, but we're trying, in order to fully recapitulate his transplant, we actually have to make an animal that's CCR5 deficient. So we're actually using CRISPR-Cas9 technology to selectively delete CCR5 alleles from our stem cell donor animals. And we have successfully done that. And we're hoping that OHSU will be the first center to have a transgenic monkey born in the United States this year.
Wow, that's going to be amazing.
Yeah.
Be written up in all the magazines and so forth.
Maybe.
Fair amount, maybe.
Maybe, but again, it's all about the science, and then, that pushes forward HIV research, but it also pushes forward a lot of gene editing for curing genetic diseases.
Right, and that's another important thing. What we learn in HIV is translatable to other areas.
Absolutely.
And it's so tied in with cancer these days.
Absolutely.
The drugs are using to cure.
And so the next thing where my lab is going now is with this allogeneic stem cell transplant model, is just understanding how can we make clinical care better for the people that are undergoing this for cancer treatment. Because having spent time in the transplant wards, I can tell you that we definitely need animal research to support a rational design of better drugs for people undergoing stem cell transplantation.
There you go. No, you're absolutely right, because it was hell what Timothy Brown went through.
Absolutely.
And I'm sure all the others were, except we were more focused at that rather than people that are otherwise unknown.
Right.
So I really appreciate your taking the time again to visit, and I hope we can do it more and more, because you're on the cutting edge.
We're close.
You are on the cutting edge. You really are.
Cheers. Thank you.
Thank you so much.